Okay, hi students, so welcome back to my video. So today we will learn a new chapter, chapter 5, Metabolisms and Enzymes. Proverbs 17 verse 22, a joyful heart is a good medicine, but a crushed spirit dries up the bones. So, we're going to study the metabolism. Okay, what is metabolism? Metabolism refers to all chemical reaction that occur in a living organism. The process in metabolism involves the conversions of food into energy in the form of ATP and the formations of carbohydrate, protein, lipid, and nucleic acid. So, we have two types of metabolism, which are catabolism and anabolism. So what is catabolism and what is anabolism? Catabolism is the process of breaking down the complex substance into simple substance. So ini adalah pemecahan complex substance kepada simpler one. Itu catabolism. Okay, so this reaction, catabolism, will release energy. Okay, for example, the breakdown of glucose, pemecahan glukos semasa sel respirasi, during cell respiration to generate energy. So these are catabolism. Okay, so these are catabolic reaction. So we have substrate. For example, substrate di sini, substrate is a bahan, the reactant lah, dia punya bahan tindak balas. Okay, so substrate here, for example, glucose, bila dia mengalami pemecahan menjadi produk, A become B plus C, this will generate energy. Okay, so ini yang kita panggil sebagai catabolism, iaitu breaking down. So remember, the keyword of catabolism is breaking down. Okay, while another types of metabolism, anabolism. This is the process of synthesizing ataupun building, okay, complex molecule from simple molecule. So from simple molecule, it will synthesize ataupun build a complex molecule. Okay, so now this reaction uses or absorbs energy. For example, the formations of glucose during photosynthesis. So this is, the reaction is reverse from the first one. Okay, here we build A plus B become C. C is the product. So this is anabolism. The keyword for anabolism is synthesizing, and it will also use ataupun absorb energy. Okay, so we go to enzyme. What is enzyme? Okay, in a cell, biochemical reactions occur at a high rate to protect the living processes. Okay, biochemical reaction can occur quickly in cells due to the presence of enzyme that help speeding up the reaction. Okay, so enzyme is an organic catalyst that is mostly made up of protein and produced by living cell organism. Okay, however, not all enzymes are synthesized from proteins. The substances needed for an enzyme reactions are called substrates. Substrates will bind with enzymes at a specific site known as the active site and form an enzyme substrate complex. So, apakah itu enzyme tadi? Enzyme ini adalah yang membantu dalam mempercepatkan satu tindak balas itu. Itu adalah enzyme. Okay, enzyme ini adalah satu pemangkin ataupun catalyst di sini yang made up of protein and dihasilkan oleh living cells organism lah. Okay, walau bagaimanapun tidak semua enzim itu dia disynthesize daripada protein. Okay, so the substance needed for an enzyme are called substrate. This is substrate. Okay, ini adalah substrate, iaitu dia punya reactant lah kan, bahan tindak balas. So the substrate ini dia akan bind to the enzyme di mana? Di specific site of the enzyme yang kita panggil sebagai active site. Active site ini adalah dia punya specific site on the enzyme where the substrate will attach to. Okay, di mana perlekatan itu berlaku, itu kita panggil active sites of the enzyme. Okay, so when substrate and enzyme dia react kan, dia akan form this one, iaitu enzyme substrate complex. Okay, so enzyme nomenclature ini adalah penamaan enzyme. In 1960s, the International Unions of Biochemistry and Molecular Biology (IUBMB) dia memperkenalkan enzyme nomenclature based on the substrate or reactions it catalyze. Okay, the name of enzyme is derived by adding ASE to the name of substrate it catalyzes. Okay, so penamaan dalam IUBMB ini, penamaan enzim itu adalah berdasarkan kepada apakah substrate yang dia mungkinkan. What is the substrate it catalyze? So and then you will add ASE. Okay, so for example here, lactase. Okay, lactase enzyme, it will catalyze the hydrolysis ataupun breakdown of lactose. So maksudnya nama enzim itu adalah berdasarkan dia punya substrate. Okay, for example here we have lactose, hydrolysis of lactose, we add water molecule, so it will be break down into glucose and galactose. So the enzyme here is lactase. So nama enzim itu adalah bergantung kepada nama substrate dia, lactose, lactase. So the OSE we change itu ASE. Okay, however there are also a few enzymes that do not follow this naming system. Okay, especially enzymes that were discovered before this systematic naming. Okay, sebelum ini dia bukan ikut seperti ini. Okay, so ada beberapa enzim juga yang tidak mengikut penamaan yang seperti ini. Contohnya adalah trypsin, pepsin, renin. Semua enzim-enzim ini adalah tidak berdasarkan substrate yang dia catalyze. Okay, so general characteristic of enzyme. Enzymes act rapidly. Okay, enzyme acts rapidly. Enzymes are only required in small quantities and are reusable. So dia hanya diperlukan dalam kuantiti yang sedikit dan boleh diguna berulang kali. The structure of enzyme remain unchanged and are not destroyed after a reaction. Okay, structure of enzyme itu dia tidak akan berubah, dia tidak akan destroy selepas satu tindak balas itu selesai, dia tidak akan destroy. Sebab itu enzim itu adalah diperlukan dalam kuantiti yang sedikit sahaja dan reusable. Okay, the reactions of enzymes are specific. Only substrate that fit the form completely with an enzyme in an active site can combine. So ingat, enzim ini adalah specific. Meaning here, okay, maksudnya di sini adalah one type of enzyme dia can catalyze one type of reaction. Begitu. Okay, so sebab enzim itu dia mempunyai active site. Okay, so only substrate that can fit with the active sites of one enzyme itu that can combine with the enzyme and then the reaction will continue. Okay, seterusnya, most of the reaction catalyzed by enzymes are reversible. Reversible maksudnya seperti ini. For example here, lactose plus water, hydrolysis, okay, catalyzed by enzyme lactase, it will be broken down into glucose and galactose. Reversible maksudnya we can reverse this reaction. Glucose plus galactose, remove one water molecule, okay, become lactose. Itu maksudnya reversible. Okay, can be like that, reversible. Okay, and then next one, enzyme activity can be slowed down or stopped by enzyme inhibitor. An examples of inhibitor is heavy metals such as lead or mercury. Okay, so maksudnya di sini Inhibitor itu adalah yang bahan yang akan memperlahankan ataupun menghentikan the actions of enzyme. So itu kita panggil inhibitor. Okay, some enzyme needs cofactor to work more efficiently. An examples of a cofactor is a vitamin B and magnesium ion. So ada beberapa enzim itu dia akan memerlukan cofactor supaya dia boleh work more efficiently. Okay, so for example, contohnya di sini dalam kehidupan seharian kita, kalau kita mau drive kereta itu perlu ada minyak. So barulah kereta itu boleh bergerak. So begitu juga dengan enzim. Okay, begitu juga dengan enzim, dia perlu cofactor supaya dia boleh work efficiently. Alright. Enzymes are biological catalysts that speed up biochemical reaction. So itu adalah enzim. Satu pemangkin biologi yang mempercepatkan biochemical reaction, tindak balas biokimia. So remember these characteristics of enzymes. So we have intracellular and extracellular enzyme. Apakah ini intra dengan extracellular enzyme? Enzyme that are synthesized in a cell for its own use are called intracellular enzyme. Okay, for example, hexokinase enzyme that is used in the glycolysis process during cell respiration. Okay. Other than that, enzyme that are secreted outside the cells are known as extracellular enzyme. For example, trypsin enzyme produced by the pancreatic cell and secreted into the duodenum to break the polypeptides. Okay, so kamu kena bezakan intra dengan extracellular. Intracellular adalah enzim yang disintesis di dalam sel dan digunakan juga di dalam sel. Tetapi extracellular ini adalah disintesis di dalam sel tetapi digunakan di luar sel. Okay. So these are the productions of extracellular enzyme. So ini adalah step bagaimana penghasilan enzim extracellular lah. Okay, the first one here. The first one, ribosome is the site of protein synthesis. So kita sudah belajar ini kan, ribosome itu apa? Ribosome itu adalah tapak sintesis protein. Okay. Protein synthesized by ribosome enter the lumens of RAF-ER and transported through it. Okay, number 1 di sini, this is ribosome. Ini ya, ribosome yang biru-biru ini. Ini ribosome. Kita tahu ribosome itu adalah tapak sintesis protein. Kita tahu bahawa enzim itu adalah protein. Okay. So protein synthesized by the ribosome will enter the RAF-ER. RAF endoplasmic reticulum and then transported through it. Okay. Kemudian step yang ketiga di sini ialah once it reaches the end of the RAF-ER, the membrane buds off to form transport vesicle. So dia akan form here transport vesicle. So the transport vesicle that contains protein will move towards the Golgi apparatus. Okay, Golgi apparatus, and then fuses with the Golgi apparatus. So in the Golgi apparatus, the proteins are modified into enzyme. Okay, protein itu modified into enzyme. Kita tahu bahawa Golgi apparatus itu dia punya keyword adalah modifying, betul kan? Okay, so dia akan modified into enzyme and then secreted in a secretory vesicles. Okay, here secretory vesicles that are formed from the tip of the Golgi. This is the Golgi apparatus tip lah. Okay, Golgi apparatus. So now the secretory vesicle here will move towards the plasma membranes of the cell. Here. Okay, and then fuse with it to secrete the enzyme out of the cell. Okay, So remember, this is the steps ataupun sequence of the productions of extracellular enzyme. From ribosome, okay, from ribosome, you're entering the rough ER, okay, and then dia akan form the transport vesicle into the Golgi apparatus. So in the Golgi apparatus, modifying, okay, protein modified into enzyme, and then secreted in secretory vesicle, okay, from the Golgi apparatus, and then moves toward the plasma membrane, and then it will secrete the extracellular enzyme out of the cell. Okay, so mechanisms of enzyme action. Okay, so remember this. If you want to explain the mechanisms of enzyme action, so remember the lock and key hypothesis. So you know the lock and key hypothesis kan? Lock itu dalam bahasa kita, lock itu dia punya mangga. Tahu kan mangga? Ini key itu dia punya kunci. So kunci dengan kita punya itu mangga. Tahu mangga atau kendadu? So ingat. So most enzymes are complex protein made up of polypeptide chains that are folded into 3D structure. The three-dimensional structure has an active site with a specific configuration that complements a specific substrate molecule. So the binding of molecule substrate on the active site of the enzyme is like lock and key. Okay, like lock and key. Here the enzyme represented by a lock, kendadu and the substrate is represented by the key. So ingat enzyme itu lock, substrate itu key, kunci. Yang enzyme itu dia punya mangga ataupun dia punya kendadu. So ingat. Number 1, the specific substrate approach the enzyme. So this is the enzyme. Okay, yang yellow ini substrate. So the substrate itu, the specific substrate approach the enzyme, okay, and then the substrate combined with the active site. Okay, remember active site of the enzyme to form enzyme substrate complex. So maksudnya substrate itu dia akan attach to the enzyme di mana di satu bahagian sahaja, iaitu pada active site-nya sahaja. Okay, so bila dia sudah combine, ini kita panggil enzyme substrate complex sudah. Okay, kemudian reaction will take place, and what happen? Products are released or produced. Okay, so then the product leaves the active sites of the enzyme after the reaction complete. Okay, so remember lock and key hypothesis. Enzyme itu macam lock lah, lock as the lock, and then the substrate is the key. So remember, kalau kita kunci pintu pun kan, only the correct key can match with the lock, betul kan? So begitu juga dengan ini enzyme action. Okay, the specific lah, specific. Okay, substrate itu kena specific dengan enzyme dia. Okay, alright. Most reaction inside the cell require high activation energy. What is activation energy? Activation energy is the energy needed to break the bond in the substrate molecule before the reaction can occur. Okay? Activation energy, tenaga pengaktifan ini adalah tenaga yang diperlukan untuk memecahkan, okay, ikatan dalam substrate molecule sebelum satu tindak balas itu boleh berlaku. Maksudnya satu tindak balas itu hanya akan berlaku sekiranya dia sudah mencapai activation energy. Selagi activation energy itu belum achieve lagi, so the reaction still cannot occur lah. Okay? So enzyme functions by lowering the activation energy. By doing so, the rate of biochemical reaction is accelerated. So maksudnya di sini, bagaimanakah enzim itu dapat mempercepatkan satu tindak balas adalah dengan cara merendahkan activation energy. So bila rendah sahaja dia punya activation energy, so kadar tindak balas itu boleh dipercepatkan. Okay, boleh dipercepatkan. Contohnya, okay, teacher bagi contoh di sini, kita ada dua orang pelajar. So CA dengan CB. Dua-dua ini mau lari memecut 100 meter. Okey, 100 meter. Di sini penghujung dia. Cumanya dia punya activation energy. Kedua-dua A, CA dengan CB ini perlu mencapai activation energy. For example, 100. Baru dia boleh start lari. Okay, activation energy dia 100. Baru boleh start lari. Okay, tetapi CA ini, dia ada enzyme. Okay, dia ada enzyme. Dia makan satu gula-gula. Gula-gula enzyme lah kan dia makan. So, bila dia makan gula-gula enzyme itu, dia punya activation energy itu lowers down, become 50. So, bila dia sudah capai the 50, dia terus lari. Lari-lari pecut-pecut sampai di penghujung garisan di sini. So, yang si B pula tadi, dia perlu capai dulu activation energy 100. Baru dia boleh start lari dan sampai ke penghujung. Siapa yang menang? Si A kah si B? Yes, si A yang menang. Sebab dia punya activation energy sudah direndahkan menjadi 50. Sebab dia ada gula-gula enzim. Ini contoh lah. Okay, ini contoh. So, begitu juga dengan enzim. Enzim itu function dia ialah by lowering the activation energy. So, when the activation energy is lowered down, so the rate of biochemical reaction can be accelerated or increase. Okay? So, kalau kita lihat di sini, yang biru ini adalah without enzyme. Yang merah ini adalah with enzyme. So, kalau kita lihat activation energy di sini, activation energy with enzyme, dia low. So, dari sini ke sini, low. Tapi activation energy without enzyme, dari sini atas sampai bawah, dia tinggi. Okay? So, maksudnya di sini, cepatlah tindak balas yang mempunyai enzyme. Itu adalah lebih cepat. Kerana enzim itu berfungsi untuk merendahkan activation energy itu. So, remember what is activation energy? Activation energy is the energy needed to be achieved by the substrate before one reaction can take place. Okay? So, belum sampai activation energy, dia belum boleh reaction itu boleh berlaku. Alright? So, the mechanisms of enzyme actions and factor changes. So, dia ada beberapa faktor yang mempengaruhi tindakan enzim lah. The first one is temperature. Kesan suhu ke atas activity enzyme. So, at low temperature, the rate of reaction catalyzed by the enzyme is low. Okay, kita tengok nombor 1 ni. Okay, this is rate of reaction here. This one is temperature. So, number 1 here, at low temperature, the rate of reaction that catalyzed by enzyme is low. So, when the temperature rises, the kinetic energy of the substrate molecules and the enzyme also increases. So, what happens when the kinetic energy increases? This will increase the frequency of effective collision. Semakin tinggi kinetic energy, semakin juga tinggi perlanggaran di antara molekul-molekul tersebut. Okay? So, what happen next is the rate of reaction between the enzyme and substrate molecules also increases. So, with every rise in temperature of 10 degrees Celsius, the rate of reaction controlled by the enzyme will be double. Okay? Until it reach the optimal temperature. Maksudnya dengan pertambahan 10 darjah Celsius, kadar tindak balas itu akan meningkat double lah. Okay, double sehingga dia mencapai suhu optimum di sini. Okay, this is optimum temperature iaitu 37. So at optimum temperature, the enzyme reaction is at the maximum. Paling tinggi lah tu rate of reaction dia. Okay, so here in human body is around 37 degrees Celsius. Okay, so after reaching the optimal temperature, Any further increase in temperature will reduce the enzyme activity. So, until it stops at 60 degree. So, pada suhu yang melebihi 40 darjah, so dia punya tindakan enzim itu akan menurun sehingga pada suhu 60 darjah dia punya activity of the enzyme will stop. Okay? Kenapa dia stop? So at this temperature the enzyme become denatured. Ternyata asli denatured. Okay, because the chemical bond in the enzyme dia sudah break. Okay, at the extreme temperature. So dia punya suhu itu untuk 60 itu sudah tinggi untuk enzyme. So chemical bond dia sudah break. Okay, sudah break. So enzyme itu unable to retain dia punya 3D shape form dia. So active site of the enzymes also they change. So the substrate cannot fit with the active sites of the enzyme. So that's why the reaction stop. Okay, the reaction stop. So dia punya optimum temperature di sini adalah 37 darjah, 37 degree Celsius. So 60 degree dia stop sudah. Because of the extreme temperature dia akan denature the enzyme. The chemical bond in the enzyme will be break. Okay, will be break down, and then dia punya shapes of the 3D shapes of the enzymes are unable to be maintained. Okay, and then the active sites of the enzyme also changes and cannot fit. Okay, cannot fit the enzyme and the substrate anymore. So that's why dia punya activity stop. Alright, remember. So next one is effect of pH on the activity of enzyme. The enzyme activity also influenced by the pH of the surrounding solution. So biasanya enzyme ini kebanyakan optimum pH dia adalah pada dia akan bertindak dengan baik pada optimum pH-nya. So most enzymes are most active in the range between 6 to 8, pH 6 dan 8. For example, salivary amylase, okay, amylase air liur, dia works the best at pH 6.8. So, however, there are some exception. There are some exception. For example, pepsin. Okay, pepsin. Enzyme pepsin ini di perut, dia act at optimal pH dia adalah between 1.5 and 2.5 here. Okay, dari sini ke sini 1.5 to 2.5 lah. Okay, and then trypsin enzyme in the duodenum. Okay, duodenum ini dalam usus, small intestine, only works well in alkaline medium. Dia suka yang alkaline punya medium. So pH dia around 8.5. So di sini 8.5. Itu trypsin. Kalau pepsin ini dia di stomach, dia suka yang acidic punya pH. Okay, tapi kebiasaannya enzyme 6 to 8 lah. For example, salivary amylase dia punya optimum pH adalah 6 and 8, begitu, 6 to 8. Alright? So, the change in pH value changes the charge H plus ion of the active sites of the enzyme and the substrate surface. As a result, the enzyme substrate complex cannot be formed. So, when the pH of the environment returns to the optimum level, the charge on the active site will also be restored. Okay? So the enzyme will return to function as normal. The extreme change in pH value will break the structural chemistry bond and change the active sites of enzyme. So it will denature the enzyme. So this is molecule of normal enzyme. So this is molecule of denatured enzyme. Okay? Next one, the effect of substrate concentration. Substrate concentration, kepekatan substrate. If the concentrations of enzyme is fixed, while the concentration of substrate is increased, the reaction rate controlled by enzyme will also increase. leading to an increase in product. So here, number 1. So this is rate of reaction and this is substrate concentration. Kepekatan substrate. When substrate concentration increase, the opportunity for an effective collision between the substrate and enzyme molecule also increase. So the reaction rate continue to increase until it reach the maximum level. Okay, the rate of reaction is constant. At the maximum level here, the concentrations of enzyme becomes a limiting factor. The reaction rate can only increase when the concentrations of enzyme increases. After reaching the maximum level, all active sites of enzymes are saturated with substrate and are involved in the catalytic reaction. So maksudnya di sini, poin utama dia adalah sekiranya enzyme itu kita fix itu enzyme concentration, kita tambah, keep increasing the concentrations of substrate. What happen? The rate of reactions also increase. Okay, tapi dia hanya sampai maximum level. Lepas itu, pada maximum level, rate of reaction dia constant, sudah constant. Kenapa constant? Sebab walaupun kita berhabis tambah substrate, berhabis tambah itu substrate, tetapi enzim kita itu juga bilangannya, concentration enzim kita kekalkan. So buat apa kita tambah substrate, berhabis tambah substrate, tapi enzim kita sudah jadi limiting factor. Maksudnya the enzyme itu sudah fully occupied by substrate. So when we add substrate again here, so the rate of reaction still constant. Okay, sebab enzyme concentration telah menjadi limiting factor. Okay, begitu. Alright. The effect of enzyme concentration. When the concentrations of enzyme increases, the rate of enzyme reaction will also increases because of the presence of more active sites that are ready for catalytic reaction. If the concentrations of enzymes in one reaction is doubled, the amount of substrate converted to product per unit of time also doubled, with the condition that there is an excess supply of substrate. So at the maximum rate here, the concentrations of substrate become the limiting factor. The rate of reaction, sini dia akan malar lah. The rate of reaction can only be increased by adding more substrate. So ini adalah sama dengan substrate concentration tadi lah. Bila kita tambah enzyme concentration, okay, bila kita tambah enzyme concentration, dia punya rate of reaction pun increase. Okay, tetapi itu sampai maximum level sahaja. Selepas maximum level, dia akan constant. Di sini rate of reaction dia constant. Sebab siapa jadi limiting factor adalah the substrate yang akan jadi limiting factor. So, kita boleh increase balik itu rate of reaction dengan cara tambah substrate lah. Okay, tambah substrate. Begitu. Alright. So, applications of enzyme in daily life. Apakah dia punya application of enzyme? So, enzyme has been widely used in commercial sector and in everyday life lah, everyday use. Okay. The enzyme use are extracted from natural resources such as bacteria or produced synthetically. Immobilized enzymes are enzymes that combine with inert and insoluble substances to increase the resistance of enzymes towards change in factors such as pH and temperature. So with this method, the enzyme molecules will remain in the same position throughout the catalytic reaction and then be separated easily from its product. So this technology is known as immobilized enzyme technology. So this technology is used in various industrial application. For example, here in medical sector, digestive enzymes are used. Okay, dalam perubatan, enzyme pencernaan digunakan. Okay, lactose enzymes are used in lactose-free milk. Pectinase and cellulase enzymes are used in juice production. Amylase, lipase, protease, and cellulase enzymes in biodetergent. Okay, trypsin enzyme extract fur from animal hide to make leather products. Protease enzyme separate the fish skin. Okay, so these are the enzyme immobilization technology used in various industrial application. Okay, so in chapter 5 you must know what is metabolism, divided into anabolism and catabolism. Anabolism is synthesize atau build up, catabolism is breaking down. So enzyme nomenclature penamaan, you add ase to the substrate. Okay, for example, lactose, lactase. Okay, general characteristic of the enzyme, okay, kena tahu. And then we have two types of enzyme, intra and extracellular enzyme. Apakah maksud intra dengan extracellular? And then the mechanisms of enzyme, okay, remember lock and key hypothesis. Dan juga factors that influence the enzyme action. Okay, so here we have temperature, pH, substrate concentration, and enzyme concentration. So applications of enzyme in daily life also. Okay, apakah kegunaan enzyme dalam kehidupan seharian? Okay, so itu sahaja untuk hari ini. Okay, a joyful heart is a good medicine, but a crushed spirit dries up the bones. Proverbs 17 verse 22. Thank you.